That's great. Hopefully everyone can lip read.
Sorry, sure.
So yeah, if you haven't already started the Nextflow repo, please can you do that for us? It will help with grant applications and that sort of stuff. We've recently submitted a CZI grant application, which we've had for a few years now, and it really helps to hire developers to help with the community and to keep things going on that front, as well as sponsoring hackathons and various other things that we're doing. Okay, so a lot of you have probably seen these famous tube map representations that actually came out of the NF4 community that are now being used everywhere. And so this is what a typical pipeline looks like. It's a series of steps that you perform in a containerized way. And Nextflow really manages a lot of the complexities behind the scenes for you, where it runs these with containers, with pre-installed, with whatever you need to run these particular tasks. But also it has a number of other features like flexibility, scalability, various other things that you can exploit in order to run these pipelines virtually anywhere. So you write a pipeline once, you do it properly, and the idea is that you can share and run that pipeline wherever you want. It has a number of features where you can write code in any language and still use that within your pipelines, as well as I mentioned, using containerization to really deal with dependencies. You know, gone are the days where you have to do an install of a tool on a HPC, and it makes it impossible to share scripts in a reproducible way, you know, across the planet if someone wants to actually get the same results. Now everything is containerized, everything's run in a container. It's a really nice, compact way of running your pipelines. But then also, I think what's really changed the way that we do science in general, in terms of bioinformatics, is version control with Git. And it sort of brought in software engineering practices into bioinformatics that weren't probably there as much before, that really force scientists to think about what they're doing with their code, how they're writing their code, how you can get others to review your code. And some of this stuff actually is also pushed us in the NF Core community to implement some of these things within the community as well. So we can exploit these features and actually start writing code in a nice, open, reproducible way. But reviewing is incredibly important. And then to top it off, you can run these pipelines virtually anyway. I think there's been some conversations and chats about that. You write a pipeline once and then run it anywhere. So very quickly, numbers, Nextflow is growing, you know, pretty much exponentially. If we look at, you know, a standard metric like how many people are accessing documentation over time, it's just going up and up. You know, 16,000 users in December, which is just phenomenal. If we look at NF Core, almost 900 contributors contributing to NF Core GitHub repositories. Again, phenomenal. If we join up Twitter followers, it's almost 9,000 for Nextflow and NF Core. The Slack organizations, we join them up, you know, 11,000 users. I joined NF Core when there were about 20, 30 people on Slack, and we were literally just building everything up at that time. And now we're almost 8,000 in the space of like 5 or 6 years. And it really sort of filled a gap for bioinformatics analysis research, not only for those that are bioinformaticians, but also those that just want to use pipelines. They don't want to have to sit there and write a complex RNA-seq pipeline. And you know, some of you in the audience here, you know, all the amazing talks that happened this morning, you write a pipeline that fills a niche. You're the domain experts, and anyone can come in and use it. And it's just amazing how it's just grown out. You can have conversations about your pipelines, various other things directly on Slack, and get help, and also, you know, stay around if possible. That's what we want. Yeah, so this is just a plot of this Slack membership over time. It generally peaks at hackathons and that sort of thing, but you know, almost 8,000 is just amazing. The Nextflow Slack now, which is a separate workspace, that we started a bit more recently. It was on Gitter before that. And even that is just, I think we're almost 2,000 members on there as well. Okay, so see, these are just some other numbers. 7K over Slack, 7K Slack users, over 40,000 commits, 16,000 pull requests, 7,000 GitHub issues. These numbers just are staggering. And then they keep on going up and up every time that we give these talks, that they're outdated. So what is the core content on NF Core? I mean, a lot of it is the pipelines. So the whole philosophy behind creating the NF Core community was that very early on, before Seqera even, at a community conference for Nextflow, a number of people decided that they would like to use Nextflow to create this resource of pipelines that were best practice pipelines that anyone can come in and use. And rather than every individual lab having their own version of an RNA-seq pipeline, they wanted to contribute and collaborate on writing a single pipeline that everyone can use. And so this is actually one of the core guidelines on NF Core, that you can only really have one pipeline per data type. And so there's only one RNA-seq pipeline, there's only one ChIP-seq pipeline, and various others. And over time now, we've really built up, it's almost like a big brain of domain expertise that we've built in this community, because I know nothing about proteomics, but others do. Yasset, we were talking to him just outside, they know about proteomics. They've contributed pipelines to NF Core. And so others that want to use these pipelines can then come in and do that. And so we're now really starting to see a shift from, just genomics type research to having proteomics pipelines, image analysis pipelines, astronomy pipelines, various other things. And in essence, anything you can containerize, you can write an Nextflow pipeline around. So it doesn't have to be even life sciences, to be honest. You can do anything in a container where you have software available. You can use Nextflow to do it and run that pipeline wherever you want. And so you can come in and just use these pipelines. And as I said, 90 pipelines. It'd be amazing to get to see us get across the 100 mark there. This is a bit what the landscape looks like. So this is a word cloud of GitHub stars across NF Core pipelines. I think this is probably outdated now, but just to give you an idea, most of the pipelines there are green because they're genomics pipelines. Then you've got red ones, which are proteomics pipelines. You've got a yellow one, which is a pangenome pipeline, which is becoming sort of the cool kid on the block when it comes to analyzing sort of human genome data. And actually, we had Adam Philippi give a keynote at the Boston Summit, which was awesome. But we're also seeing a lot more image analysis pipelines. Image analysis is a completely different type of data because it's huge, or it can be huge. And you can't open it on a laptop to start identifying cells and circling them. It's virtually impossible. And so what you need to do is you need to be able to use good containerized software to take that image analysis to a series of steps. It's also very difficult to move around. It gives you a different problem to running 20,000 small tasks. This is like running, a few huge tasks. And, how do you provision the storage? How do you run that, whether it's in the cloud or locally on a HPC? It's a completely different problem. And so this is what the landscape looks like. They've also, as I said, they've been the sizes here represent the number of stars per repo. And so the RNA-seq pipeline is clearly the most used pipeline, it seems, the most starred. And that's mostly also because there's just a ton of RNA-seq data knocking about. A lot of people do RNA-seq experiments. And so it's there. It's also probably the gold standard NF Core pipeline that we've got. Everything that we sort of implement and develop on RNA-seq, it tends to be done there first as a proof of concept to ensure that it's working before we roll it out more widely on NF Core. These are the typical components of a pipeline by our definition on NF Core. When I say our, I mean with my NF Core hat. That's so a community type hat, not necessarily a Seqera one, even though they're quite interchangeable. So here a pipeline is an end-to-end pipeline that runs some sort of analysis. A sub workflow is just a series of steps or modules that we call them that allow you to chain together some tasks. So doing some adapter trimming and then running FastQC after that, and then maybe doing something else after that. So you've got like 3 or 4 components that form a sub workflow. These sub workflows are also amenable to be shared across pipelines. And so the smaller and more compact you can make these to make them shareable, it just means that you can more efficiently implement your pipelines at scale. And then underneath that, we've got modules. So a sub workflow is formed of a series of modules. A module is a single unit in our definition of Nextflow, which is just a single process. And that would be a process to run, say, BWA-MEM or FastQC and so on. And the fact that these are all individual, again, it just means that it makes it a lot easier to share. I mean, we had to come up with a way of really adopting DSL2 syntax of Nextflow on NF Core. And this is what we eventually ended up with, is having a repository of core modules that we can have in a single repo. Contributors come and contribute those, update them, and also be able to install them in any NF Core pipeline, any Nextflow pipeline as well. So over time, we've come up with some standards for how we implement sub-workflows. They're sitting in the NF Core modules repository. We've got 54 of those. And now we've got almost 1,100 modules on NF Core, which is, again, it's just amazing. Lots of people are coming in, contributing these modules for their own pipelines. They're reusing them, they're updating them, they're identifying bugs and issues and fixes and pushing them back. We're also updating the tooling and various other things on the NF Core front to keep them up to date and to make sure that we're at the edge of using all of the best sort of software implementations and practices and that sort of stuff over time. So I started this repository maybe 3 or 4 years ago at ISMB in Basel, and it just sat there doing nothing for a year. And then Paolo pushed out DSL2, and we decided we had to do something about it. And so we first implemented a prototype DSL2 implementation for RNA-seq. With some horrible syntax we used in that, but it worked. And then over time, it's just evolved and got better and better and better. And now, you know, we're sort of at 1,100 modules, which is, again, it's just phenomenal. So everything I've been talking to you about has been mostly what we're doing on NF Core, but there's also a lot of usage outside of NF Core. And there are a couple of talks today, actually, also where, you know, you guys are developing your own pipelines and you're using the template, or you're using the tooling, or you're using various other components because you just identified that it's easier to use and manage your pipelines in that way. And you can also adhere to some sort of standard internally, so you don't have to make your own up. And that, again, it's just amazing seeing those talks this morning and how you're using all of these things. And so if you do a quick search of GitHub, for example, and look for some things that are quite unique to the NF Core pipeline template, there's over 1,000 pipelines on GitHub that are using this. And this is just GitHub. This doesn't include Bitbucket and GitLab and all of the others that are out there as well. And so actually, just following on from that, what we're trying to do also, as I'll probably tell you a bit later on in future directions, is to make the NF Core template more accessible, simpler, easier to share. It's been commonly requested across the community. You know, people want to get the NF Core goodness, but they don't necessarily want all of the boilerplate, and that's understandable. And so what we're trying to do is to try and make that a bit more fine-grained and tunable. And so whenever you update or create your pipeline, you can select what you'd like to do. So it will be more opt-in and easier for you to then build on top of that and start writing your own Nextflow pipelines. And so I think that's a work in progress right now. So NF Core pipelines also, you know, are being used everywhere for reproducible research. And that's one of the main advantages. Science has to be reproducible. Otherwise, there's no point doing it, really. And so NF Core pipelines are being used in all sorts of papers. And I think the thing that I love the most about this is the fact that when you write your methods section, it can just be a single line. So I've used this pipeline with this version, potentially some parameters, and that's it, right? Because everything behind the scenes is actually managed by containers and various other things that are already pre-bundled with that version of the software. Before NF Core, you know, I was having to reproduce public data analysis, and it can be a nightmare. You have no idea how someone's run a pipeline, what parameters they've used. It's very light on the details. And so trying to reproduce those plots, summaries to write your own publication at that time, you know, was horrible. And this sort of helps with that quite a lot, providing you've used the same command and have access to the same input data, which most publications now ask you to do anyway to submit to public databases. You can reproduce what you've done and hand it off to the scientists to actually focus on what they want to add as the rest of their publication, which is just phenomenal. Okay, so the tooling. As I mentioned, we've got some guidelines. These are almost sort of the 10 commandments of NF Core, and we have some standards, some guidelines that we implemented and wrote, and they're evolving, to be honest. But the idea is that it really helps to set some sort of spine in terms of how users in the community interact and build their own pipelines. And I think it's really nice to have those. Otherwise, you end up with loose definitions of things, and there's not enough clarity on what you need to be doing and how you need to be doing them. And so we were very strict about these standards initially, and that was for a reason. But actually, very few people have complained about them. They see the standards, and they're like, okay, well, we want to write our pipelines in this way as well, because they see the benefits, and it's awesome to be able to see that in person. So I guess one of the key ones is, as I mentioned, not having more than one pipeline per data type, but also the way that your pipeline is written. It comes with standard test data. It's containerized, and so that it can work on virtually any platform if you like. It's tested on virtual platforms. We've got CI/CD as well, which is also potentially sort of a guideline that your pipeline works before release via CI/CD. Ideally on clouds as well. We've got credits from cloud providers because they are also seeing massive usage of NF Core pipelines on all these different clouds because they're just easy to use. And so I guess following on from that, that's another thing that NF Core has really changed is a lot of people are transitioning to cloud, and they have been doing that over the past few years. Big pharma, small pharma, academia, all of them are transitioning to the cloud. And inherently, Nextflow and NF Core pipelines, in the way that they're written, work on all these cloud providers, which is great because now companies are moving to the cloud and they're like, well, I need an RNA-seq pipeline. Where am I going to get that from? And so they look at NF Core and they're like, okay, we can just use this out of the box. And so now we're seeing a massive shift where all of this open source contribution now is paying off in the way that it's changing the mindset in the private industry as well, because they now also want to contribute back to open source rather than having everything gated off, which is typically, how potentially things were done at that time, rewriting their own pipelines, having their own bioinformatics and that sort of thing. Now we're seeing our customers wanting us to contribute back to open source and allowing us to do that, which is absolutely phenomenal. Okay, so in terms of how you use what we've got on NF Core, there's pipelines, there's sub-workflows, modules. You can start stitching your own pipelines together. There's various linting options as well, so that we know that the template and the pipeline you're writing is adhering to best practices. There's tooling and also this parameter schema that allows you to update your pipelines. But behind the scenes, a lot of what's going on NF Core is via this NF Core tools package. The reason we need that is because we don't want to be going to every 90 pipeline and submitting a pull request to update one portion of the template. It doesn't make sense. So the only real way that we can attempt to automate a lot of what we're trying to do across 90 or 100 pipelines is to have some sort of tooling that we implement and we developed. And so we've got this NF Core tools Python package, and it allows you to do various things, whether that's creating a pipeline template or creating a sub-workflow template or creating a module template or linting or downloading containers because you want to run in an offline environment. All of this is pushed to this NF Core tools package. Whenever NF Core tools is released on pip, what happens is NF Core tools itself contains a skeleton pipeline template. Behind the scenes, what happens is when NF Core tools is released, there's a sync mechanism that gets triggered off, and it automatically creates a pull request to 90 pipelines in the same go to now update that pipeline to the latest version of that template. And so this way we can stay up to date. Whenever NF Core tools is released, we have one point to update and it gets rolled out across all of these different pipelines. And it's just a really nice, easy way of interacting with these things. But there's various other commands here as well that I'll show you in a sec. Wave was mentioned earlier. And actually, we do have a pull request on NF Core modules to add CI/CD to actually build those mod containers via Wave. We just need to figure out some of the details. It's very important to get things right. A big advocate of doing things in a small way and then rolling it out on a larger scale, because then you can try and iron out a lot of the issues that you're seeing there. But I guess one of the key things that potentially also we need to think about in the NF Core community is how are we environmentally responsible for some of the stuff that we're pushing out in NF Core. It's all well and good building 1,000 containers, but that has an environment impact as well. And so how can we be better with our CI/CD? How can we be better with the size of containers? So, for example, what Wave does behind the scenes is when you have a Conda recipe in your Nextflow process, what it will do is it will, and you decide you want to build that container with Wave, what it will do is it will use a micromamba base image, and it will essentially just do a Conda env create behind the scenes that you don't see within a Docker file. And that Docker file then gets pushed up all transparently via Wave, and then you can use that. And so the idea would be then on NF Core, we could do something similar. Most modules have a Conda definition. Why can't we just use Wave for that? But I think we need to figure out how we do that in a sustainable way. So we've been discussing potentially having our own registry where we can push up these containers, but also doing a bit more profiling of the size of these containers. Are we able to make these containers smaller? If you think about how many times the SAMTools container is pulled globally, maybe even just down to NF Core pipelines, it's probably some phenomenal number, thousands, tens of thousands. And if we can shrink the size of that container by, I don't know, 20, 30 megs, whatever it is, it makes a massive difference running these, especially in the cloud, because every task you run in the cloud has to be in its own virtual machine. And so for each of those tasks, you have to pull that container locally before you can run anything. And so I think we just need to be a bit more aware about how we're having an impact on the environment to not be wasteful, essentially. And also part of that has been also thought about in the implementation of the NF test that we've been doing that I'll talk about in a second. So you can install this. It's available on pip, NF Core tools. If you just do a help, there are various options there, not only for users, but also for developers there that you can go and have a play around with. So we've intentionally started to now make a shift in the way that we're developing our tooling so that it also supports custom pipelines. As I mentioned, and as we saw this morning, there are custom pipelines that are being implemented and developed with NF Core tooling, whether they're on GitHub, Bitbucket, GitLab. And so part of that is adding the tooling that supports all of this. So for example, you could have a completely custom pipeline with all NF Core components, or you can shift out and just have a few sub workflows that you want to install from NF Core and maybe a module or two. Or you could have a completely custom pipeline. You could have your own modules repository, your own RIP mirror of that, and the tooling itself should resolve that and install what you need within your pipeline. And we know it's being used, but we haven't had much feedback. So if you are doing this and you've got any issues, then please let us know. We'd love to, have some proper use cases where we can implement and improve some of the stuff that's going on here. But it's entirely possible to do things this way. There's also widespread adoption of the tooling and various other things that we're doing and advocacy, in fact. So Backtopia, for example, it's a huge pipeline that's just written by someone at Robert Petit, who gave a talk at the Boston Summit. And it's got tons of components, and it's his own custom pipeline, but he's still using a lot of NF Core modules and contributing back those modules for everyone else to use, as well as sub workflows. There's a Tree of Life project here that we know is, using NF Core components in their own implementations. ONT are using the schema that we developed on NF Core and various others. And Element Bioscience is also a massive advocate and also are quite heavily invested in the NF4 demultiplexing pipeline for their platform. Right, so NF test. There's so way back when we started implementing NF core modules, we needed a way to test that the modules worked. There's no point adding 100 modules to a repository that don't work, because then you have no idea whether when you install it in the pipeline, it's just going to break the pipeline. And so we needed a way to test those. We needed unit testing. And so PyTest workflow at that time solved the problem. It was great. You could write standard YAML, and then you can update and implement that. Someone creates a pull request, the CI/CD is automatically triggered off on the pull request. So whenever someone makes an update to that module, if it breaks, then you know you have to fix it. And so it's a really easy way for users to get onboarded, not only just bioinformaticians, but also core users that want to write a module and are just starting to write their Nextflow pipelines. Is a fairly easy way to keep on top of it. But it has its own limitations, and it's not Nextflow native. So for example, you can't test functions and various other things that are written in Nextflow or in Groovy. And NF tests came about, can't remember when it was now, a year or 2 years ago, maybe, where they really sort of announced it. And very recently, we've been working with the authors of NF tests who have been really receptive and have taken on our feedback and suggestions and implemented a bunch of stuff in NF tests, which is great. And what this means is now we can start using NF tests as our testing framework. There is a transition going on at the moment where we're shifting from PyTest workflow to NF test. There was a massive effort at the hackathon, and there will be, I suspect, over the course of the next year or two, shifting that. But not only within modules, but also within pipelines. And so at the moment where we've come up with sort of what a skeleton pipeline would look like with NF test in it, and we're just figuring out now how we do it. But a very key thing, going back to my point about environmental impact, is if we have NF tests in all of the components in your pipeline. So right now, the only test that we include in NF core pipelines is the end-to-end pipeline test, essentially, that runs all of the processes to make sure it's working. But if we can ship all of the tests that the pipeline performs by default, whether that's the modules, the sub-workflows, and the workflows, we can start doing clever stuff with CI/CD where we can essentially check for changes. So we only run the tests that have changed. Rather than rerunning everything all of the time, we can be a bit more selective about what we're running. And again, it just means that, you know, someone's not paying the light bill, and also we're being a bit kinder to planet Earth. So this is still a work in progress. The hope is to have an RNA-seq release out soon-ish with that. A lot of my team at Seqera, as well as in the community, have been working on pushing all of these NF tests to the components that are being used in RNA-seq so we can get a proof of concept together. Fetch NGS actually has already been released with a lot of the core concepts that are being used in NF tests. So yeah, worth keeping an eye out. There are various other plugins. The NF validation plugin actually also came from NF Core. We developed JSON schema to represent parameters that are used in pipelines, and now it's being used all over the place for various things. Help text generation, pattern validation, sample sheet validation, and it's a really powerful way of just keeping on top of the parameters that you're using in your pipeline. Also, because you can now start rendering these JSON schemas in user interfaces. So at the Epitomean Oxford Nanopore platform, that's what they're using to render their particular user interface. And that's what we're using also on the Seqera platform as well, to render that platform, to make it easy and open up the way that people run Nextflow pipelines. You don't have to run Nextflow pipelines via the command line anymore. You can now start using user interfaces to run them. It's your choice whether you want to use command line or whether you want to use a user interface, or whether you just want to trigger them automatically. You don't need to know. Data hits a bucket and you just want to trigger a series of events that then gives you some sort of data at the end that you're interested in. The NF Core website has been built from the ground up again. Matthias has been working around the clock on this, and now we've got a brand new NF Core website, which is a bit more scalable and removes a lot of the hidden gems that Phil had in the old website. So yeah, that's awesome. In terms of the tooling, we've recently improved the download capability, and We've added also partially as a result of this, a new Nextflow command called Nextflow Inspect, which allows you to look at the containers that are in a pipeline. We had some horrible regex code that was parsing Nextflow scripts that is incredibly fragile, and I wouldn't ask anyone to even look at what that regex looked like. But we needed something a bit more Nextflow native. And now we've got that with this new Nextflow Inspect command. It's also compatible with Wave, and there's various other features that you can get in there as well. There's also a user-friendly terminal app that Phil has been helping to write, which brings this new tool called Textual in, and essentially it's called a TUI. So rather than a user interface, it's a terminal user interface. And so rather than typing out your command, you get prompted via terminal type interface to enter things within there. And again, it's a really nice way of interacting with the terminal. Okay, so very quickly on community initiatives. There's a podcast, if you didn't know already, called Channels. And I think these run every few weeks or something like that. It's worth keeping an eye on these. There's always sort of new content and new discussions around various things that are happening in the Nextflow world, and new features and various other things that we're adding maybe also to the NF Core community as well. We have a new ambassadors program on the Nextflow side. Priyanka's part of that, and she's done an awesome job organizing this, got us all here, which is amazing. And so if you're interested in becoming an ambassador, you can find the details on the Nextflow website. We also have a new community forum. So the plan is eventually to move Nextflow and even multi-QC and various other things into this discourse type format. And the main advantage of this is that it now allows you to Google for things, which is huge. Everyone Googles for things. In Slack, you can't necessarily do that. But also there's an advantage to using something like Discourse because it allows you to scale. And given the size of these communities, as I showed you earlier, we need something that is actually resilient to that. And so we're trying to come up with a unified way of actually communicating. And so please check it out. It's I think it's community.seqera.io or something like that. There's more details. Also, Phil's talk at the summit in Boston, which is on YouTube now, goes into this in a bit more detail. But it's really nice to be able to do this and move things out there. Also events, so talks like this, bite-sized seminars, training. So Geraldine Van Oera, who was at the Broad at the GATK, developing and helping out a lot with the GATK best practices, has now joined Seqera as lead developer advocate. Phil has moved a bit more into sort of open source platform type of role within Seqera, and so she's now going to be driving a lot of these efforts here to start engaging with community and seeing how we could help, training, various other things that are really important. And lastly, thank you for listening, and thank you for inviting me to speak.
We've got a bit of time, so do we have any questions? Priyanka.
Okay, so moving forward in terms of, how would you say people who are coming from Nextflow to NF Core can start? What's like the first step to contributing to NF Core? How can they be part of the community in their learning cycle?
I mean, it's probably not the answer you want, but just ask lots of questions. Don't be afraid to ask questions. Everyone at some point, including me, was completely, head spinning about what's going on and how to do things. But if you ask questions and are interested, then we've got all of these community channels where you can come in, join the community Slack, join the Nextflow Slack, join various other places that are useful to you. And we've also got tons of documentation as well online that you can follow, you can get up to scratch with. If the documentation isn't right, please let us know, because the fact that you're a beginner means, I almost think that beginners write the best documentation because they have to go through that process themselves, and writing it down in a nice, easy, understandable way is priceless for me. And so if you're confused about anything, ask questions, but join the community first. Each pipeline has its own channel on the NF Core Slack, for example, and you can join there, ask questions about, you know, how you'd like to get involved, any questions about the pipeline. There are various other channels there as well.
Yeah.
So you said there are another duplicates of pipeline. You have only one pipeline for people and sequence. I think it's good, and it makes you some sort of gatekeeper. You have a great responsibility. My question is how do you ensure that the pipeline that you unique pipelines having the best tools that can possibly have, because they are browsing for some of the.
So the best way, again, is just to have this conversation in Slack with the pipeline developers. Some pipelines will be outdated. You know, a lot of, I mean, I didn't say, but you know, a lot of people contributing to NF Core, it's complete voluntary contributions. And so they all have day jobs as well, whether they're using these pipelines for their own research or whether they're using them or contributing back because they've got some funding within the lab to contribute, or they're working as part of a wider organization like BovReg or the Tree of Life. And they need to, you know, improve the tooling and the pipelines in some way. What I would say in that case is just ask on Slack. If you are comfortable adding that, I mean, again, this is the big advantage of having an open community, is that these pipelines are sitting there. They may not be up to date, but you can always contribute as well. You know, push issues, ask questions, create pull requests, and the pipeline improves over time. So in general, what gets added is a community decision. And in order to make that decision, you need to have a conversation somewhere, and Slack is probably the best place to do it. If we can get contributions, then that's a massive bonus back out of it.